Good afternoon, everybody. Hope all is well. Hope all is well. It was a nice afternoon, and it's turning out to be a beautiful day here in the big ATL. All righty. We were working on our worksheet, but we're not going to do that in class today because we're moving on. But you will have time to do some work. Just wait for a few more people to come in. But we are here on this page, DNA mutations. So prior to that, the page that I'm talking about, like I said, in spite of the fact that I told all of you that you could have had your worksheet done by the end of class, only half of you did. But please make sure you get this worksheet turned in. I do want to say something about this one. And it's going to be the same thing on the one that you're going to do today. Technically, we should be translating this RNA strand twice because this RNA should be messenger RNA and then translated to transfer RNA, which is the one that actually picks up the amino acids. Okay, so because of that, on this first row, you ended up with two stop codons, which is not really supposed to happen. Okay, so you had a start and a stop. All right, but make sure this worksheet gets turned in. Like I said, some of you did, some of you didn't, but we're moving on. Alrighty. So take a minute. We're going to talk about mutations today. There are different types of mutations. So take a minute to be in Canvas. I'll give it another minute. One more minute. Somebody else is still missing. And let me say this, they've already put out the exam schedule, which means this is the week before Thanksgiving. So we're off next week. And then when you come back, you got a week and a half, basically, to finish up this unit. And then we're done for the semester. So some of you who are falling behind need to make sure that you are turning stuff in. Like that worksheet should have been completed in one class period. Shouldn't be something that you're still trying to work on, because we have another one to do today. So please, 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 please try to stay up and turning in all of your assignments on time. All right. If you turn something in on Friday, I will check as soon as we are working on your independent work. All right. So, all right. If you look at this box, this one has that there are different types of mutations. So the video that we're going to watch deals with that. And then we'll come back to this. All righty. And I've already put it in on the playlist. So we're going to watch it from the playlist. Because I think when you watch it from the playlist, it takes out all those commercials. Or some of them anyway. Oh,
Your personal finances start with you, but they support your entire home team. With Thrivent, your money can be a force
Get bigger.
For good for the people, causes, and communities you're most thankful for. Thrivent, where money means more.
They would determine if we will watch that.
Ever since I was a kid, I have been curious about mutations. I think certain movies may have had an impact. I used to imagine mutations with amazing abilities, but there was also a lot I didn't understand about mutations. Let me explain. Many people understand that a mutation is a change of genetic material.
Please pay
More
attention,
specifically,
this
a
is
change within a
more
nucleic
complicated
acid.
than the last.
RNA and DNA are both types of nucleic acids. Therefore, anything with RNA or DNA can have a mutation. That means animals, and that definitely includes humans, also plants, fungi, protists, bacteria, archaea, they can all have mutations. Additionally, so can viruses. Many mutations can be neutral in effect. You can see this codon codes for the amino acid leucine. But if it experiences this particular silent mutation, even though a base has been altered, it still codes for leucine. It did not change the amino acid. Mutations can also be harmful or helpful, but it's important to understand that mutations are random. The organism can't will itself to get a certain mutation. You can learn more about this in our natural selection video, where we talk about how a bacterium may already possess a helpful mutation that allows it to survive an antibiotic. But the bacterium didn't will itself to mutate to develop this certain mutation. Okay, so again, mutations are random. But there can be factors that make mutations more likely to occur. External factors like certain types of chemicals or excessive radiation, or internal factors like an event that causes a problem during DNA replication in interphase. During interphase, cells can replicate DNA before they divide. So let's discuss some different types of mutations. We'll start with gene mutations. DNA makes up genes, and genes can code for proteins that influence different traits. So when a mutation in DNA happens, which specifically means a change in one or more DNA bases, then different proteins can be produced, which can affect an organism's traits. In this example, we have fruit fly DNA. Mutations could include substitution, which means the wrong base is matched. Insertion, which means an extra base or bases are added in. There is also deletion, which means a base is removed.
All right, I'm going to pause it here just to, so these are 3 vocabulary words. We're going to be working with those as soon as we come off the video. Those are, we're going to give our attention to these types of mutations.
Insertions and deletions have the potential to be especially dangerous. Why? Remember in protein synthesis how we talked about how bases are read in threes? Well, if you add a base or remove a base, suddenly the number of bases total has changed. And if you read the bases in threes, depending on
Remember,
where it
you should be on
happened,
camera.
everything that is read afterwards could be affected. We call this a frameshift mutation. If you look at this frameshift example here, where a single base has been inserted, you can see how the following codons, which have 3 bases each, are now all affected as the reading frame has been shifted. This example shows how it can lead to many amino acid changes. There are also chromosomal mutations. Remember that chromosomes are made up of DNA and protein, highly organized, and they have lots of genes on them. The human chromosome number is 46, with 23 from an egg cell and 23 from a sperm cell. This type of fruit fly here, however, has 8 chromosomes, so 4 came from an egg cell and 4 came from a sperm cell. Some examples of chromosomal mutations include duplication, where extra copies of genes are generated, deletion, where some of the genetic material breaks off, inversion, when a broken chromosome segment gets inversed, which means reversed, and put back on the chromosome, or translocation, when a fragment from one chromosome breaks off and attaches to another chromosome. There's more mutations than what we covered, of course, but the idea is that there are many different kinds of changes that can happen. If a mutation is going to happen, we already mentioned that there are especially vulnerable times, such as during DNA replication, but also there are other times too, like during meiosis. In fruit flies and other animals, meiosis makes sperm and egg cells that can have half the number of chromosomes as the organism. However, sometimes those chromosomes don't separate completely. We call this non-disjunction. This can result in an egg or sperm cell that has too many or too few chromosomes. It's possible for a mutation to be passed down to offspring. Consider a protist with a mutation. Many protists reproduce asexually, and when they divide, the daughter cell can inherit the same mutation. A fruit fly, which reproduces sexually, can pass a mutation to its offspring if that mutation is found in the genetic material of the sperm or egg cell. You may wonder why we've been mentioning fruit flies so much. Well, they're awesome, but also it turns out that fruit fly mutations and how they are inherited are frequently studied. We have some links in the video description for more info. These studied mutations can also occur in humans, and we'll give a gene mutation example of a substitution in the case of sickle cell anemia. First, a little background. Hemoglobin is a protein in your red blood cells that helps you carry oxygen. But in the disorder sickle cell anemia, the gene that codes for hemoglobin is mutated. If you inherit 2 copies of this gene, one from each parent, you can have this disorder. This disorder can make it difficult for your red blood cells to carry oxygen because the shape of the red blood cell is affected from this mutated hemoglobin protein. This can lead to anemia and other problems. While unfortunately there is not a cure for this disorder yet, the good news is that treatment for this disorder has greatly improved. Another thing to mention, if an individual only inherits 1 copy of the mutated gene from one parent, they are a carrier, but they don't officially have the disease. Usually they do not have symptoms, but those that are carriers appear to have a protective factor against malaria. Malaria is a disease caused by a protist that can be transmitted by mosquitoes. These individuals can still get malaria, but their symptoms are often less severe. We should mention that studying mutations and genetic disorders is a large and important field right now. Genetic counselors work to help families that may be affected by genetic disorders. If you have an interest in learning more about careers related to this topic, please check out some of our further reading suggestions in the description below. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, so several of you came in late. That video is in your playlist. It's nice to be back off of probation, so I can save stuff to your playlist again. All right, so let's get back to the module. All right, so we're in DNA lessons and mutations. So we looked at last week how to create a protein. The DNA is going to open up. The messenger RNA is going to read the information. It's going to go to the ribosome, and it's going to collect amino acids and create a string of amino acids, and it makes a protein. And we learned how to read the chart. We're going to use this chart again, and we're going to see how when you have one normal strand of DNA or RNA, and when you have an abnormal strand of RNA, what happens, what it creates. Oops. This is just a real short blip. All right, but the important part about mutations, it's an error in copying the code during replication. So in other words, if sometimes when you read a sentence and you leave out a word, sometimes it doesn't affect what you're understanding, and sometimes it could change the whole understanding of what you have just read. Same thing with messenger RNA and translating the DNA message. The DNA message is telling you one thing, and the messenger RNA skips one or puts too many together, or it deletes a whole section. And so when it goes to make the protein, the transfer RNA reads what it brings it, but it's missing something. So these missing things can cause it to translate an error. Okay. So they are random. Most of the time they're minor, they don't bother you. Some can be deadly, some cause disease. Very few are helpful. Okay, so let's look at All right, so again, it's mutation is random, where the sequence of amino acids is altered. And any single amino acids that is wrong in the wrong place can cause an enzyme to malfunction. And the malfunction will cause a disease such as sickle cell. So you have different kinds. Okay, you have random, and then actually this is what they already said. Mutagens. It's anything that causes a mutation. So sometimes it's not that it is read wrong. It is sometimes um, something in the environment that causes a change in your genetic information. So like you've been exposed to ultraviolet radiation, smoking, or asbestos. Those are things that can cause changes in your chromosomes. And if you change the chromosome, it changes the way the DNA is read. All right, let's look at this one. Okay, so then you've got three different kinds. So you've got a missense, where the base pair results in a different amino acid, and it may not affect the protein. For example, we had when we looked at our chart, several of the amino acids can be coded in different ways. So if it changes it in a way where you're still creating the same amino acid, that's not bad. Okay, then you have some that are nonsense. They don't make any sense. The base pair changes and the result means a premature stop codon. So the protein is non-functional, which means we had three different ways that we could stop the protein from making. And so if it doesn't read the whole message, and it's, have you ever played telephone and you try to get that message from one end of the one person has to relate the same message all the way down to the 20th person, and by the time it gets to the end, it's not the same one message that we started with. That's what happens with nonsense. You get a message, but something's missing. It's shorter, it's longer, or it just doesn't make any sense at all. That's what happens with that. Silent type of genetic disorder. The base pair results in the same amino acid, and there's no change to the protein. Okay. All right, so we're going to look at it on our worksheet. Then there's a frame shift. So remember, these amino acids are read in groups of 3. So say it starts reading it in a place where it wasn't supposed to start. So that first triplet code is not the one it should have started with. Then you have a shift in the message. You can do this by adding, deleting of the nitrogen base, like the A, T, or G, or U. You can add one, delete one, and it'll change the way, it'll shift the way the message is read. Okay. And then one we just talked about. And we'll talk about this one some more later, because we're probably when we come back in January, because we have to do a whole section on mitosis and meiosis, like she mentioned in the video. That's a whole unit in itself. All right, so we'll talk more about this type of mutation, but for today, we're going to talk mostly about gene mutations. All right, so what I want you to do, like I said, we'll come back to that. What I want you to do is download this worksheet, and I'll get you started on it. And then, all right, so it's in the module. It's the very next thing. If you download the worksheet, it's going to look like, and I'm going to give you a minute to download it, read it over. Whoops. All right, it's taking a minute, but it'll be there. So I'm going to close and come, but I'm going to let you download it, and I'll come back and download mine in a minute. I'm going to do attendance, and I'll be right back. And if it takes a little longer, just give it some time. It'll download. But look it over, and then the two boxes at the top, I'm gonna help you to decode that. So you need your little chart. And what I do is you can't see, I keep a chart up where I can see it so that I'm not flipping the worksheet. 'Cause there is a chart with the worksheet, but it's way at the bottom. So that you don't have to keep going back and forth, I would have a copy of this off to the side. Because you're gonna need it. Okay. Like I said, quite a few of you were late today, so make sure that you go back and watch the video from the beginning. All right, everybody should have their worksheet. I'm looking at folks. This is the end of this semester. You should have your worksheet up and not all over the place. I said I'll be right back. I should be looking at Go Guardian, and everybody's worksheet should be visible. Still got some of y'all, and I have no sympathy for late assignments when you're not doing your work in class. So if you want to play games, like I said, some of you didn't turn in the last worksheet, and now we're working on the second one. Okay. Let me see if mine will load now. It's had a minute. Because by now you should have read the paragraph. All right. And I saw somebody was working on the last worksheet. Stop what you're doing now and work on this one first. And if you go after I finish explaining what you need to do, then you can go back and make up what you need to make up. Uh, And in a minute, I'm just going to pull it up from my file. All right. I'm just going to pull it up from my file. Okay. You should have a worksheet that. There we go. All right, mine was taking forever. Is there anybody else? Okay, I'll check on it, Ethan. I'll get it. All right, but I need everybody's attention on this right now. Everybody should have a worksheet up that looks like this. Anybody not able to get that worksheet to come up? I know mine was delaying. I don't think you're going to have that trouble. All right, everybody should have a worksheet up that looks like this. Anybody not able to get that worksheet to come up? I know mine was delaying. I don't think you're going to have that trouble. All right, Okay. All right, so we have 2 rows, so similar to the worksheet that you had before. You've got 2 rows of DNA. So and there got one is a human strand and one is from a cow, both mammals. The first one, so it says use the codon table, and that's this. And you're going to make a complementary strand of RNA, and then you're going to write down, create a write the names of the amino acid sequence below. And right now we're not asking you to name it. We just want you to write the amino acid sequence. Okay. All right. So that means that this one is going to be your transfer RNA. Okay. So the first box is CC. So if you got your chart, and again, you're going to read the first letter is on the side. The second letter, you go across the top. The third letter, you go down the side. And actually, by the time you get to the third letter, you should just be able to find the box that you're in and find the third one within that box. All right, so we have CCA is our first one. So I go on my chart. C is going to be the second row. And the next one is also a C. So I go from the top, it's the second column. So second row, second column is that box that says proline. So it doesn't matter what the third letter is, even though it is A. It's going to be proline. So what we're looking for, I'm sorry, hold on. I did that backwards. Stop. First thing we have to do is translate it. Okay, so that first row, we want to say C is matching up with G. The second C is also a G because we're trying to create an RNA strand. Now remember, A does with T, but there's no T's in RNA, so that has to be a U. Okay, so in that first box, you're going to have GGU. And let's just go across the whole thing. I'm gonna do both the cow and the human with you. All right, the second one is TAG. T always goes with A, so we can put an A there. But again, the A goes with T, but there's no T's in RNA, so that's going to be a U. And the G goes with C. So our next box should be AUC. All right. Third box is CAC. So we go on the chart. And our C is always going to match up with G. And I saw somebody, I don't know whether it was this class or my other class, they put their complementary pairs on the worksheet so they don't forget. Because you do need to remember. Because last time somebody says, how do you know? You know because you're supposed to know. It's just something that you have to say. I always know that G goes with C and A goes with T. Unless it's an RNA, then it goes with U. Okay? T always goes with A. So even on an RNA strand, a T will translate to an A, but an A will always translate to a U on the RNA strand. So we got an A on our DNA, it's going to translate to U. And our C is going to translate to G. So our third box is GUG. All right, somebody type in the chat what the fourth box is. I might just call on you. Make sure you're paying attention.
CAA.
All right, CAA. All right. Next box in French, just Keone cannot answer. Somebody else. Isaiah. What's the fifth box? I'm gonna start calling on you. What's the fifth box? ACA is going to translate to what? Okay. And he's not talking. He put it in the chat. And folks, when you put stuff in the chat, make sure you send it to everybody. Okay, and Jocelyn said it too. Thank you, Jocelyn. All right. All right, next one. Somebody other than Keone and Isaiah and Jocelyn. Somebody else. Tell me what the next one is. ACG. What are we going to have? So you can type it in the chat or come off mute. Just say so. And see, this part is important because if you get this part wrong, the next part will be wrong also. We'll have a mutation. Okay. Come on, folks. Akari, y'all are here. Pay attention. Participate. What is ACG going to be? And again, if you don't want to call it out, just type it in the chat. Charlie, what's TGA? I'm going to move down the line again. And Neve, you tell me what AGG is going to be. All right, so first we need ACA. Come on, folks. Y'all holding up class because you don't want to participate. Brandon, you can get the last one. People. If you don't practice, you can't get it right. Y'all can just sit there and watch and copy off of everybody else, and you won't know how to do it yourself. All right, Brandon, he did the last one. Thank you. It is you. So the AAA is the last one. That's you. All right, anybody else want to? So let's go back. What is our ACA? Anybody answer it? Because we're going to move on. Those of y'all who don't want to participate. All right, that's okay. Thank you, Brandon. Okay, that's UGU. And Ethan, make sure you send it to everybody and not just me, but thank you. It's all right. So it's UGU, and then our ACG is going to be translated to UGC. Thank you, Jocelyn. And our our TGA. Okay, ACU. Thank you. And then our AGG. AGG is going to be UCC. Thank you very much. All right, and Brandon already told us that the last one is going to be UUU. All right, so now your job is to take that information and go to the chart and figure out what amino acids you're going to. So here you're going to put the initials. So I had already done the first one for you incorrectly. So we want G. It's the last row, and then go up to the top. G again, that's the fourth column. So we know we're in the last row, fourth column, and then we come all the way down to find the U, and it's going to code for glycine. So you can write GLY, and that'll be sufficient. All right, then we go to the next box, and we're looking for AUC. So we start on the right with A, that's our third row. U is the first row, so we're third row, first column, and the last one is C. So that's going to be leucine. Okay, hold on 1 second. All right, you all have a worksheet, so I'm going to come out of this for a sec and let you all look at. Mine's still ain't coming up. Let me try it again. Try one more time. By now it should be up. Okay, never mind. We're not gonna wait on that. All right, I'm going to come back over here because make this one really big. This chart has all of the amino acids spelled out for you. Okay, there we go. All right, so that's actually ILE for that first one. ILE. So that's isolene, isoleucine.
Wait, the first one is ILE.
No, the first one was GLY. The
Oh, I
second
thought
one.
it's because you said the first one.
Yeah, I'm sorry. I'm sorry. The first one is glycine. But this one, because of the way they printed, L's and I's look similar. So the first is it's ILE, which is isoleucine. Okay. I can't. All righty. Okay. Now where are we? All right. So then we have GUG. So again, last row, fourth column, come all the way down to the bottom, and that's going to be GLY again. Because there's 4 different ways to get that amino acid. But they're all going to code for glycine.
Wait, isn't that Val?
You got G.
Isn't it G?
Yes, you're right. You're right. It's G. You're so good. Thank you. It's GUG. And I was doing GG. I was doing it backwards. Okay. Yes, it is. It's GUG, which is Val. Thank you for staying on top. All right, CCA. So C, second row. A, third row, through a third column, and A again. So this one is GLN. And GLN is glutamic acid. Right? Okay. I wish this wasn't so fuzzy. Okay. Next one. UGU. U. First row, last column. And it's at the top of that box. And that's going to be CYS, which is cysteine. Then we have UGC, which is also the same. That's 2 different ways to get that same amino acid. Next one, ACU, third row, second column. And again, there's 4 different ways to get THR, which is threonine. Some of these words are so wonderful. THR. All right, UCC. first rows, second column. And again, all code, 4 ways to code for serine. And last, U. All right, so top row, first column, and the top box, U. So that's PHE. Okay. All right. I'm going to let you all do the next one. And then after you've completed those two, then you can answer the analysis questions. They want you to compare your two strands, and they want you to circle the base that are different in the cow and the human. And they want you to say how many bases are different. Okay. Number 2 wants you to highlight the amino acid produced. And so what you're doing for number 1 is you're doing the row for the RNA. For number 2, you're highlighting the third row with the amino acid. And then you're answering the question for number 3. So everybody be working on that right now. And then we'll look, and the rest of the worksheet deals with the different types of mutation. Point mutation, silent mutation. They give the example of sickle cell. Shift mutation, and they give the example of cystic fibrosis. And lastly, synthesis. Muscular dystrophy. So you may have to do a little research on some of these. So let me come out of this giant screen. Do you need me to leave this up? No. When they, okay, let me come out. They mean the bases, like the which codes are different. Okay, it starts, the worksheet starts off by saying that the strand of DNA that they pulled out is, and I had my picture up. Where'd it go? Okay, I'll just open it again. Okay, it starts off by telling you that both the strand for the cow and the strand for the human are coding for the protein that produces insulin. So what you're looking at is, how are they different? How are they the same? If they're both coding for insulin, they should be similar, okay? Because insulin is a similar product in mammals. Okay, so this is the point of the question. When you create your RNA strand, is there a difference in the triplet code in the cow or the human? That's you're going to circle. Then you're going to come down to the third row and highlight whether the amino acid you made is different than the one in the human, or are they the same? Okay, so that's what you're trying to do. All right, again, the point is they're both making the same thing. They're both coding for insulin. But is it different in a cow than it is in a human? I'm sorry, the number 1 wants you to look, okay, I'll tell you, number 1 wants you to look at the DNA. So right now I'm looking at the second triplet in the DNA, and they're different. On the DNA, on the human, it's got TAG. On the cow, it's AAG. The next one is in the human, it's CAC. In the cow, it's CAT. And if you go down a few more, we have TGA in the human, we have CGA in the cow, and then the last one, you have AAA in the human and GAC on the cow. So those should be circled on both strands.
Would we write 3 or 6 of how many there are different?
I have 4.
You have 4?
I have on the second one, the third one, and then
No, I think the third one is the same.
on the DNA strand, on the human DNA, you have TAG. On
T on the human DNA?
the human DNA, the second one is TAG, and on the cow, it's AAG. The third
Yeah.
one excuse me, is CAC, but on the cow, it's CAT. So that's 2 right there. And then if you go down, skip 3. All right, that's 4, 5, 6. The seventh one, it's TGA on the human, but on the cow, it's CGA. So that's the third one. And then the last one is AAA on the human, and it's GAC on the cow. So that's 4. Okay. That's the DNA strand. All right, so then you translate all of those, and then you want to look at what amino acids were produced. You want to highlight those and then say how many of those are different, or are they coding for the same amino acid? So number 1, you're circling the DNA. Number 2, you're looking at, are they giving you, even though they're different codes, are they giving you, are you, are they giving you this different amino acids? Are we straight, everybody? For example, even though this third one was CAT, which was different than the other one, they both gave you the same code for valine. So that amino acid is not different. It's the same. So we wouldn't highlight that. Oops. Actually, there's just one. No, two. I just highlighted the wrong. There's two. Make sure you highlight the correct two. And I'm going to tell you the answer for number 3 is yes, but you need to tell me why. All right, so when you finish with that, one of the types of mutation, and we might just only get through this page today. One of the types of mutation is called point mutation, diabetes. And you're going to need to remember which diseases are which types. So diabetes is a disease that is characterized as a point mutation. It's the ability to break down sugar, which is what our insulin situation is. Often a person with diabetes has a defective DNA sequence that codes for making the insulin protein. This mutation is called a point mutation because only one base is affected. When 2 amino acids in a sequence have been changed, the shape of the protein changes and becomes less functional or non-functional at all. So this is one of those mutations that's not good. Some, but not all types of diabetes are caused by a point mutation. So if you want to do more research on diabetes, you can click on this link and it'll tell you more about diabetes. All right, suppose a person has a so we're now using the set that we just had because we just did our chain for insulin. Suppose a person has a mutation in their DNA and the first triplet code, and now we're dealing with the human, the first triplet code for insulin is TAT instead of the CCA that you have on your chart. Determine what amino acid you'll get next. So that's the first thing you have to do. You have to translate, make an RNA strand with that translated from TAT. So somebody tell me what that is. Type that in the chat. What's TAT going to translate to? What's the triplet code? So that's a DNA. TAT is the DNA. What RNA are we going to get from?
ILE.
Well, hold on. First, you got to give me the triplet code. So what do we? So what's the triplet code going to be?
AUA.
AUA. And so he went on the chart and he found that that was ILE. Okay, that's correct. So this is going to be a change. So it made a different type of amino acid on your chart that you first did in at the top. The first one was supposed to be glycine. And now it's coding. Our first one is coding for something else. So yes. How do you know? because it's coding for a different amino acid, therefore the protein will be different. Okay, it's like spelling your name wrong. All right, so I just gave you the answer to number 4. All right. So that was if you changed all 3 letters in the triplet code. Now, again, if we look back at our chart from the beginning, our triplet code was CCA. So question number 5 is talking about a silent mutation. When one of the nucleotides changes, that means one letter is changing, not the triplet. Okay, resulting in the same amino acid. The protein that is made from this new DNA will have no functional difference than the original. So what they're giving you, so go ahead and look up CCG. And what is CCG going to give you? First you need to tell me what the
It would
triplet,
be GGC and it'd be glycine.
which is the same thing, correct? If you compare
Yeah.
it to your chart at the top, the first one that you made at the top. And we're going to keep comparing it to the charts at the top, folks. All right, so when we first made our strand of DNA, we started off with CCA, and it translated to GGU, and it made glycine. Now they're asking you to look at CCG, and CCG translates to GGC. If you look at your chart, GGC also translates to glycine. So we haven't changed the amino acid, we just changed how we got it. So the important thing is the end product. What protein are we making? Are we making the protein that we need to make, or is it trying to make something else? If you make something else, it can't do its job. Okay? And insulin is anybody have diabetes? Any of y'all diabetic? You know anything about diabetes? No, you're all healthy. That's good. That's good. All right, so we only got about 2 minutes left. So we've just done number 4 and 5. So make sure you complete those. So next class, we're going to look at sickle cell. Okay. All righty. I'm going to stop right there. And so have this handy on Wednesday, and we'll keep on going. Any questions before we go? Okay. And again, Tuesday afternoon, 1:30 to 2, tutorial. If you need help, if you just didn't understand, didn't want to talk up in class. Thanks to those of you who participated. Makes it seem like I'm not talking to myself sometimes. All right, you all have a great afternoon, and see you on Wednesday.